TerraForms. It's a process developed by EMSL earth scientist Arunima Bhattacharjee and biologist Jade Alfrick to create a synthetic plant habitat to reproduce mineralogy on a small chip you can hold in the palm of your hand. It's something almost out of a movie. And today, Bhattacharjee tells us how using TerraForms can help us learn more about various climate and environmental issues. So first, I just want to say thank you for joining me and being on EMSL's podcast.
Yeah, it's a pleasure. I'm super excited. Always happy to share more information about TerraForms.
Start off by just introducing yourself and telling me a little bit about your research and the path to your research.
So I'm an earth scientist, and I've been at EMSL now for 5 years, a little over 5 years. And so I was hired on the soils SFA as a postdoc, and I started working with soil, specifically with soil microbes, to see how in different moisture conditions, microbes grow in soil and degrade carbon, and specifically to use imaging mass spectrometry. So looking at using different types of chemical imaging to understand the metabolite distribution as these microbes grow around these carbon substrates. Just wanted to make it clear that before I started my postdoc here, I was working on all medically relevant bacteria. So coming in here, it was very different experience because I was working with one or two microbes, which are very model microbes, I would say. And then as I started working with soil, it is super complex. And that's the reason why I wanted to develop these different platforms where we can specifically do chemical imaging on them. And yeah, it was very hard to visualize soil, I would say, because it's so opaque and heterogeneous. So we wanted to develop something where we retain the heterogeneity, because the heterogeneity is super important. It is why we have these huge biodiversity of microbes, and which we love to study. And so how can we retain this biodiversity and still be able to, visualize them not only through optical imaging, just looking at them growing using a microscope, but also to get some chemical information spatially. What kind of metabolites they're making as they're growing around inside of soil pores, while they're interacting with each other, and while they're degrading minerals or degrading different carbon sources. So all this information is important because once we understand them, we can build better systems for different agricultural practices. And so that's basically my research.
You mentioned mass spectrometry. What are some other ways that you take a look at these metabolites?
So definitely mass spectrometry based techniques are primarily used. And within that, we use MALDI, we use nanodesi, we use SIMS, nanosims, and all of these. So what I have realized is not one technique can be used to decipher the whole process of, nutrient cycling, for example, what kind of metabolites they're making, what metabolites chelate certain inorganic nutrients, uptake them inside of their, inside the hyphae in case of fungus, or bacteria inside of the bacteria cell. So we need a combination of several different techniques, and which led us to make these TerraForm platforms compatible for a wide range of analysis techniques. So just to give a summary, mass spectrometry, which includes all the techniques that I talked about, MALDI, nanodesi, SIMS, TOF SIMS, nanosims, we can do SEM on them to visualize what kind of changes are happening on the surface of the minerals, how the fungal hyphae is growing within these soil pores. We have not done a transmission electron microscopy on them because you need a very thin substrate. We have collaboration with SSRL, where we use different synchrotron-based techniques like XANES and X-ray fluorescence to understand the chemical changes occurring within these minerals, the chemistry of different inorganic nutrients being uptaken by these microorganisms.
Very cool. I want to hone in a little bit on the TerraForms. It sounds really cool, and I know that that's kind of a rebrand for these micro models. Can you explain to someone who doesn't know what are TerraForms?
Just as a very basic view, if you think about it, it's like these are very reduced complexity platforms. The reason for terraforms is soil is super complex. If you have done studies in soil, you always end up with like certain conclusions, which is not very satisfactory. You can always say this thing influences this, or a group of microbes did something, but what is that? We don't completely understand the mechanisms. So once you have a hypothesis, when while studying soil, and you want to study that hypothesis further, you go into these reduced complexity platforms. The reason I say reduced complexity is because you can start building certain elements of soil into these platforms. You can mimic the soil porosity, you can mimic certain types of aggregate size distributions, you can mimic mineralogy. You don't even have to mimic everything. You can mimic only one or two of these components and then incubate microorganisms, incubate plants, by which I mean you can grow plants, you can grow microbes, you can see how they interact within these platforms and test specific hypotheses with certain soil elements. That's basically what terraforms are. And we have different platforms for users who are interested in studying plants, who are interested in studying only microbes, maybe plant microbe, or if they want high throughput, we have platforms for that as well.
What kind of hypotheses are being tested through these complex terraforms?
Different users come in with different types of hypotheses. We have one user who's looking at suppose there are different freeze thaw cycles. In case of like permafrost soils, you have these freeze thaw events. And so what happens when you have like freeze thaw cycles going on? And then how does like certain inorganic nutrients attach to certain porosities or certain aggregates or certain minerals? So looking at like how mineralogy affects plant and microbe growth, what kind of metabolite distribution occurs when you have certain types of minerals present, and what happens when you have like certain microbes growing inside of these platforms and interacting with these with plant roots. So yeah, it's a wide range of hypotheses.
And with that said, what kind of issues are we trying to solve in this research?
Well, we typically accept user proposals that aligns with the EMSL mission. This particular capability fits within the rhizosphere sensor, IRP, very well. And also, to a certain extent, the biogeochemical transformation when we talk about a lot of soil minerals. The issues that we are trying to solve is like, suppose you have very nutrient impoverished soils. So phosphorus is a very limiting nutrient right now. And so a lot of user proposals that we get look at how can we increase the availability of phosphorus for plant growth. That's one thing. Potassium is a macronutrient also, which is very limiting in certain soils. So how can we improve crop productivity? I guess that's one big issue. The other issue that we look at using these using the terraforms platforms are a carbon sequestration on minerals, for example. How can we increase that? So because there's like so much climate change happening right now, there is a big driver to sequester the carbon from the environment. And so understanding these mechanisms of like how carbon is retained on mineral surfaces, I think is really important. So I think these are some very hot topics that users come to research through user proposals and to access this capability.
Now, in some of my research, I understand there's a bit of a demand for these types of capabilities right now. Can you explain why?
I think it's because of the reduced complexity of these platforms. So, just to make it clear, this is not like the first time that anybody has done work on inside of like these tiny platforms. Well, it starts with microfluidics, and microfluidics is like not very new. It's been around for like decades in biomedical engineering. In the case of soil ecology, however, it has become more and more something that users want and people are using. So when I was like starting my work as a postdoc, I looked into this, and I think the first ever instance of micromodel use was something like going back to like 2014, 15, something like that. The papers had started appearing there. There are researchers at Europe who use these kind of micromodels. There is our collaborator at University of Connecticut who has done a lot of research on micromodels growing in platforms which has these soil aggregate structures and things like that. So optical micro EcoFab, for example, from Berkeley Lab is one example where they have done so much ring trials, including labs, and also asking labs to use these platforms. And if you kind of look back on these, a lot of these platforms are only compatible with optical microscopy. They can extract the bulk biomass and do some omics, but our advantage is that all of our platforms are compatible with imaging mass spectrometry. So we can take a chemical image on these after the incubations are done. Most of these techniques are destructive. We are trying to figure out how we can, in the future, do non-destructive chemical imaging on these. We're not there yet. But the reason why there is a huge user demand is I think it's because we can get chemical maps, and that chemical information and the metabolomic information is extremely valuable for understanding mechanisms and interaction pathways between plant microbe or microbe-microbe interaction, apart from just getting a optical image.
Right. And I'm curious if it's easier to create this synthetic version of these habitats as opposed to doing just straight field work.
I don't know about that. I'm not a very field work person. I have worked with soil in the past, but someone else obtained the soil for me. It is certainly easier to incubate inside of these devices. You're able to do like some very basic research. You can really look at mechanisms, but I don't think that these devices should be just used by themselves. I think these should be used concurrently with the existing techniques to study soil. Only then we can get a full picture. By themselves, I don't think they mean they you I mean they give you a lot of information, but it's not the full picture.
Which is very similar to a lot of research through EMSL. And for those who haven't visited, we have a really impressive plant lab, and I've seen those rhizochips with the roots growing through them. How do you start the process of growing these terraforms?
First, there is the process of fabrication itself, which we do in the EMSL clean room. And the first thing is to fabricate these devices. It is a process called photolithography, through which we create these chips. And we use different materials. We can use PDMS, cover these with membranes, which are compatible with mass spectrometry. We also use different types of epoxies, which are compatible with mass spectrometry. And it depends on what the user wants or what the project requires. If we want to embed minerals, then we typically use different types of polymers to create to, I guess, simulate the mineralogy that is required for certain projects. All of these devices are then mass spec compatible at the end. And then a seedling is grown from a seed. It is germinated on agar plates. And once it is big enough to be transferred, it is transferred into the chip directly, where it continues growing. The chip itself is filled with media and is maintained in a hydrated condition so that the seedling can keep growing. And so certain projects require addition of microbes. So that is done later on. We have different ports created inside of the device so that like we can add microbes if required. One of the projects that was coming in, I guess I'm not sure if it is funded yet, but that requested this particular platform was to look at plant, bacteria, and fungi interaction and how minerals are uptaken by fungi or bacteria and transferred to the plant, for example. So yeah, that's the whole process. And once the incubation is done, the chip is lyophilized and prepped for mass spec. But while the growth is occurring, it can be imaged by microscopy to track growth.
Before we wrap up, can you explain why this research is important to helping solve various environmental issues?
Well, primarily, again, going back to the idea of reduced complexity of these platforms is why we are able to understand mechanisms of nutrient uptake. If you have a super complex environment such as soil, then there are too many variables and you cannot really understand which variable influences how. And so I guess like this is the biggest advantage where we can have only the influence of certain variables and understand specific mechanisms, which help us understand the process at a broader level. I think that's why.
I also want to give you the opportunity to add anything else that you think we should know.
Well, I know this is a super interesting and exciting capability, and there is a lot of demand. Fabricating these devices is still something that we still do it in house. That's something we are not training users, but we would like to train users how to prep their own samples so that we can ship these chips to their respective labs. They can do their own incubation and growth, and then prep samples and send it back to us for mass spec. And if they want to know this, I encourage people to register for the user meeting and come for these workshops where you can teach them.
Now, can you tell us where we can learn more about you and your research?
It's in the EMSL website. I do have papers published. A lot of these micromodel papers are coming out right now.
Well, thank you so much for joining me today on Bonding Over Science.
Yeah, it's so great because I'm super excited as well, and this capability is something that has seen great user demand, and I'm very happy because this is something that came out from my own research, and it's really great to see people using it.
This episode was recorded prior to the EMSL user meeting workshop. If you're interested in listening to the EMSL user meeting recordings, you can find them on the EMSL website under emsl.pannl.gov/learn/user-meeting and learn more about research developed at EMSL that's benefiting users around the world. Thank you for listening to Bonding Over Science. I'm Dawn Stringer for the Environmental Molecular Sciences Laboratory. We don't have time to cover it all, so don't forget to check out emsl.pannl.gov for a full article on this topic featuring who I spoke with today. And don't forget to follow us on all social media platforms for the latest and greatest news coming from EMSL. EMSL is a Department of Energy Office of Science national user facility that accelerates scientific discovery and pioneers new capabilities to understand biological and environmental processes across temporal and spatial scales. EMSL leads the scientific community toward a predictive understanding of complex biological and environmental systems to enable sustainable solutions to the nation's energy and environmental challenges. If you're interested in working with EMSL, learn more at emsl.pannl.gov. That's emsl.pannl.gov.